Good morning, everyone. Today is my great pleasure to introduce Dr. Leluo Guan as our guest speaker for the special seminar of the National Center for Livestock and the Environment here at the U of M. Dr. Guan is a Tier 1 Canada researcher and professor in the Faculty of Land and Food Systems at the University of British Columbia. She's also a professor emeritus in the Department of Agriculture, Food and Nutritional Sciences at the University of Alberta. Dr. Guan is one of the pioneer researchers in studying the contribution of the rumen microbiome to physiology and performance of cattle, and her research program focuses on many different aspects, including interconnection of the gut microbiome and cattle genome using functional genomics and advanced microbiology techniques such as metagenomic and metatranscriptomics. She has also done extensive research on the association of the gut microbiome with feed efficiency, methane emission, meat and milk quality and safety, as well as gut development and health in both beef and dairy cattle. Most recently, Dr. Guan's group have started exploring the role of specific genetic traits, such as regulatory RNAs, to better understand the impact of key production and health traits of the cattle. Dr. Guan is the author and co-author of over 290 peer-reviewed publications and 8 book chapters with an H index of over 80. She has secured over $65 million in funding with over $25 million as a PI, as the main PI, and we all know in this room that what those numbers means. Yeah. But aside from these outstanding academic achievements, Dr. Guan is also an exemplary mentor that supports the career development of her students and many of the young scientists. I personally feel very grateful and privileged to have her as my mentor. She's also currently an adjunct professor in our department. She's contributing to the training and advisory committee of many of the grad students here at the U of M. And with that, I want to ask everyone to please join welcoming Dr. Guan to our department, and the floor is yours, Dr. Guan. Thank you. Thanks for joining.
Thank you, Human, for very nice introduction. I don't know what to say, but first I would like to say thank you for the department, give an opportunity to work with you, Keith and Kim in the past many years, and it's my actually really great pleasure to come here to enjoy the nice sunny weather. And at the moment, Winnipeg is way better than Vancouver because it was pouring before I came, and cold, pouring, wet. When I see the sun, I really wish, I say, oh, maybe I should live in Winnipeg rather than this very beautiful UBC campus. So Human has already introduced what I do, so I'm not going to go through details, but I want to give you some ideas. What I'm going to talk today is after so many years in fundamental microbiome research, looking at genes, expression, regulatory mechanism, I actually really start to think about how we potentially use those knowledge potential, we can translate them into the technologies that farmers can adopt. So I'll start with, it's funny, when I yesterday prepared the slide, I see there's lots of graduate students here, so I thought about a journey, how to think about find novel research that determine a way that you can be a potential leader. So this is me, I would say almost like 25 years ago when I came to Canada. And then when I start to look for a future research career in my life, we're not going to compare, how young I was, but it's a good way that to see how to potentially translate a lab research into industry-related applications. So this is our first idea, is how to link the rumen microbiome to feed efficiency at the time that rumen microbiology and feed efficiency and the genetics are separated. And it is really novel at the time we thought about to merge them or put on how potentially rumen microbiome contribute to feed efficiency. And that's how, oops, now it's not working. The Zoom is working, but now I can see myself, but the slide is not working. On how potentially we can link the rumen microbiome into the feed efficiency. So if you look at the ideas, well, just maybe when we figure out the technology, I want to give you some ideas to our graduate students, is because it took me actually 3 years to publish this very small manuscript. And when it was published, I received reviews, comments, 5 of them, 23 pages, because at the time, they say, no, we don't believe that rumen microbiome is directly linked to the feed efficiency. And it took me, I sent to the Journal of Animal Science, I get 3 major revisions, and then later editor says, so no, it does not feed the vision of the journal. I sent to a lot of journals, they just continue to criticize this idea. However, right now you can see there is a very common now, people include rumen microbiome in many feed cattle trials to determine how the microbiome can play a role to affecting host. Be aware, we always need to be aware that microbiome is not everything. Microbiome is just part of a puzzle or part of a picture. So host can potentially have most of the control in many traits. So we always need to think about, when we determine microbiome's impact, we really need to consider what host may play a role in affecting those traits. So through those many years, so what we are working on right now, so what I want to talk today is what are our main research directions, how to potentially translate research, the microbiome fundamental functional genomics research into adoptable technology solutions for industry. So I list these 4 areas or directions we're working on. So you can see here, I use the novel microbiome solutions, meaning we can potentially manipulate, intervene microbiome to improve its efficiency in terms of fiber, carbohydrate digestion, nutrient generation, microbial protein synthesis, and so on. And also how we can potentially select the microbiome as a target so we can use for potentially breeding and so on. So So one of the first idea, I'm just going to show the project we are working on. So this is a project also has a Manitoba kind of involvement. You can see here, we have an industry partner from MSP, Starch Products, Inc., that actually worked with us trying to look at potential probiotics we isolated from calves, and together with those probiotics, we use those potato-resistant starch manufactured in Manitoba, and we actually conduct calf trials. So those project was supported by Alberta Funding Consortium, RADA, Alberta Milk, and Saskatchewan Milk. So we actually isolated probiotics from the neonatal calves. We then also created a model that calves, they don't receive enough colostrum, likely they will have a failure immunity passive transfer, and then it is speculated they will have higher sustainability to the disease. So then we give them the probiotics, prebiotics, and the symbiotics. Now we are working on the mechanisms. Because this trial was done during a very difficult time, and we actually can only put the calves in the vitro one by one, and it was a lot of challenges. So the results mainly showing we didn't see any changes in diarrhea growth because all the calves were managed in the vitro, but we do see the calves fed with bifidobacterium or the symbiotics, they actually improve their lung score. So I don't have time to introduce the data, but we now look at mechanisms, how potentially the probiotics, bifidobacterium, and the symbiotics, which the prebiotics and the probiotics can improve the lung function. So hopefully next time when I come here, I can show some of the results. The second area we're working on is identify novel effective products, enzymes from the rumen that we can potentially use the enzyme as a feed additives to improve the digestive efficiency using low fiber forages. I really appreciate, you can see here, Dr. Kim Omniski. At the time, we collaborate, and we actually published 4 papers together, but this is one of the main contributions that we actually used the rumen samples collected at Beef Center at University of Manitoba. We actually were able to use a novel approach to discover novel enzymes. So what is the novel approach what we used? Lots of research now for microbiome, we do metagenome sequencing. But we all know, even we have the genes, just like human, we all potentially carry genes that lead to cancer. But those genes may not express, right? So for rumen microbiome, we can see lots of genes using metagenome, but those are only genetic potential. And microbes, they do not express those genes all the time. This is one of the key challenges when we try to purify, isolate novel enzymes from the rumen system. So what we did, we actually used the metatranscriptome, the RNA level, so those genes are already expressed. Then we can go back to use those RNA transcripts to predict its protein structure, and then we isolate those proteins and also determine their structure and the function. So in this study, we actually collect the rumen samples 4 time points. We can potentially look at when the bulls are fed high forage diet, how the because it's high forage, high fiber, how the transcripts that involved in carbohydrate active or metabolism enzymes were expressed. So we were able to actually found candidates, and we were able to, based on the transcripts, we know the enzymes were expressed, we can predict their protein structure, and then we were able to express proteins using protein expression vector. We can potentially look at their biochemical kinetics and also compare to commercial available enzymes. So this enzyme 1 is a xylanase, and we use the sigma xylanase extracted as a commercial product to compare. We found actually this enzyme has better activity than the commercial available sigma enzymes. So since then, we are working on other 14 enzymes. Those are the 13, and we also found another one from YAC metatranscriptome. And currently, we are actually testing those enzymes using in vitro assays. And you can see here, there is a couple of other enzyme candidates actually had way higher activity than the enzyme 1. So this project has been, continually supported by RADA and Alberta Milk and also Saskatchewan Cattle Association. And even recently, so BCRC also continuing support this project because we have been collaborating with Dr. Tim McAllister. We are continuing using in vitro assays to determine or select potentially most effective enzymes, and potentially we can use them for animal trials. You may ask why we spent so much time to trying to find enzymes. One of the challenge for all the feed additives enzyme from commercial company is they are not original from cattle. Many of them are through yeast, fermentation, or found from paper or, wood, kind of a degradation, lignin or cellulose. So when those enzymes are not endogenous, they are exogenous. In many cases, they cannot be functional into a rumen environment. So the idea is we discover enzymes from the origin of the rumen, and then we put it back to the rumen. It is speculated it should be more effective. So based on Dr. Tim McAllister's collaboration research, we do see the enzyme 3 actually has very high function when it was incubated with a barley straw compared to the rumen fluid inoculation. So hopefully we will, by certain time, we'll find potentially novel enzymes that can be adopted by the industry. The third part is about breeding. So this is actually one of my personal interests. The research question behind is, microbes is always in animal or in the rumen, in human like our body. So why they don't overgrow, there's always nutrient rich. There must be a host mechanisms regulate the microorganisms. But if there is a host mechanisms, what could be? And also the question is, can host or animal select microbes? So this is the idea behind. So lead to the one of the first publication to identify that potentially rumen microbes could be heritable. And we also identified rumen microbes that are associated with specific genome types. Here mentioned, you can see here the SNP, single nucleotide polymorphism. And in this particular research, we found that particular rumen microbial taxa are highly associated with host. Here we use the word moderate heritability. Be aware, this is actually not a true heritability. It's actually estimated heritability using genome-wide association study, meaning GWAS. So since we published, now you can see lots of study now start to include host genome typing together with rumen microbiome into the research design to see whether there are certain rumen microbes could be potentially selected or associated with by host. So why this is important? So give you an example, if the microbes are selected by the host and they actually contribute highly in terms of high methane, then what we need to do, right? We know they were selected by host. We know they contribute high methane. So we might need to find a way, in the beginning, we don't breed cattle with those rumen microbes. So we can actually use the genetic breeding, select microbes, they contribute to the low methane or high feed efficiency. So this is the idea how potentially we can use those kind of a research that we can breed animals with desired rumen microbes. So if we can find certain microbes that are highly associated, selected by host, they can contribute high feed efficiency, low methane, we can actually select them, breed them. So in a way, we can have more option for the industry to breed cattle with a more efficient and a low methane emission. So this is the table I want to show you. I know it's complicated, just ignore. I just want to know what we are working on. So at the time, we actually identified rumen microbes are highly associated with the host, but also we went to the QTL database. We found this specific SNP is actually known to be associated with average daily gain, dry matter intake, and MCR. So as a researcher, I'm always curious why. And then I would just want to show you why we are focused on this gene, because this gene is very interesting. It actually involves in potassium channel. And one of the reasons I thought about, there's one research we never do about rumen microbiome is rumen movement, right? We know cattle, the rumen actually move. Any of the students, while you collect rumen samples from cannulate cows, you put your arm in, what you feel? Movement, right? Pressure. And so this is mean what? This is the question. Does the rumen movement actually affecting microbial efficiency? And does the rumen microbiome movement potentially select specific rumen microbes that to make them more or less efficient? We do see this gene expressed in our rumen tissue epithelial RNA-seq data set. Now we are actually genome typing animals with this specific gene to potentially identify whether they can select specific rumen microbes. So as I mentioned before, the heritability we published is more like estimate. So then some people always ask, how do you know they were truly heritable? You are just doing association. So then we start to think about, what about mother and daughter? What about different generations? So we started to look at dairy, because beef is difficult. Most of beef steers are slaughtered. We cannot really identify a group of cohort as offspring. So in collaboration with my previous students, who is now a professor in the Zhejiang University in China, so we actually look at dairy cow. We can see the cows and heifers or the different generation. And what we found that actually the certain microbes from the mother cows can be passed to the offspring. And what interestingly is, when we look at the microbes passed from mother to the daughter, the heritable host-associated ones are more stable compared to the non-heritable ones, the host-non-associated ones. So suggesting there will be a potential pathway mechanism that rumen microbes can be passed from mother to the daughter. So this is another area I'm very interested. I'm continuing to understand how potentially genetics can contribute to the rumen microbial population, taxa, and their function. So the last part, I'd like to move on to the current, one of a very large consortium, a project we are working on. I just want to introduce this project, especially you can see here, Human is one of the co-lead, which play very important role in this project. So this project is funded by UNSEC Sustainable Agriculture Initiative Program. And we have partners from different universities. So UBC, University of Alberta, University of Guelph, University of Manitoba, University of Saskatchewan. We also have our government partner and the private partner from CMAX, LockToNet. We also have an international partner from United States, which is, I am part of a Green Cattle Initiative, and also the INRA-led Horizon H2020 Holoruminant project. So for this particular project, our goal is potentially to deliver adoptable solutions for our industry to improve the cattle feed efficiency and reduce the methane. And this project also is co-funded by many industry partners, and including BCRC, RADA, Alberta beef producers, and also, as I said, I'm part of a Green Cattle Initiative, and because Canada, this Green Cattle Initiative actually is in collaboration with Dr. Tim McAllister and Stephanie Terry. We are actually direct get funding from the Green Cattle Initiative to potentially understand what contribute to the project overall in North America. So for this particular project, this is always the question, right? What are the best approach to mitigate methane? So this is my postdoc summarized this very slide, basically to look at up to date the approaches or strategies that can be used to mitigate methane. So you can see here, there is a diet manipulation and plant breeding, which is really novel idea, animal breeding. But also this is where we are working on with Green Cattle Initiative to understand hydrogen sink. And also there are certain people looking at the feed management, how they potentially affect methane emission. But there's all sorts of large scale life cycle analysis, farm management. But also we can talk about animal management as well as the rumen microbiome. So based on those ideas in the past, we worked through Together Canada. Sorry, it's very wordy, but I just want to follow what we propose in this large funded program. So the objective for this particular project is one, to establish the integrated database for the rumen microbiome that link to the feed efficiency methane emission from both dairy and beef cattle, and also to develop a novel machine learning to determine the causal effect of the microbes, and then to also understand host genetic effect, potentially to develop a estimate the breeding value for rumen microbiome contribution to methane. And then the other most important is led by human, is develop one of the first culture collection at Canada. Potentially we can use combined with Objective 1 to formulate microbial consortia, microbial direct-fed microbes. Potentially we can use as a feed additive to improve feed efficiency and to reduce methane. Of course, in this project, we have a really group of researchers in social science, economics. They support our research on to potentially identify what are the barriers industry will adopt the technology and how potentially assist for policy making and so on. So for this particular project, sorry, you can see it's very wordy. We have different activities, and within each activity, we have a lot of research groups, programs. And we can see here, there is research on generating large rumen microbiome data set, machine learning development, and also we have a biobank of microbes, but also there are fundamental manipulation technologies. I will introduce a few of them. And then, of course, validate our previous research on the heritability of the rumen microbiome and potentially to identify what is the best approach, G times E, about genetics and the nutrition management to potentially manipulate the rumen microbiome. And then we will potentially have the validation study that we can potentially use for different strategies. So I'll re-emphasize this again, but the idea is we want to potentially develop a 3R strategy that we can use the right genetics for right microbiome intervention and for the right management. Hope, after 4 years, we can deliver the goal. So I want to think about this, how we are going to work. So potentially we are going to look at the rumen samples from the grazing beef cows and feedlot cattle, breeding cows, as well as growing and the lactating cows, and potentially to look at the heritable and high feed efficiency, low methane, and how we can potentially lead to the breeding potential. And also non-heritable and high feed efficiency rumen microbes, we can use them as a feed additives to feed to those heritable and high feed, low feed efficiency or high methane, or as together with those non-heritable ones and so on. So basically our goal is potentially to develop a different strategy to improve feed efficiency and lower the methane. So this is our goal, and I hope we can all work together. As you can see, I didn't have time to introduce all the co-investigators from different organizations. We have researchers from AFC, 4 research centers in Lacombe, in Sherbrooke, and in Lethbridge. And we also have, researchers heavily involved in economics and different cattle trials. But we aim to work together potentially to reduce the methane with this estimate number, average 37 point, which hopefully with the goal to reduce the cost for the production of the milk and meat, and also contribute to the environment sustainability. So with the rest maybe of 10 minutes, quickly I'll talk about what we do at the UBC. So I mainly lead on the project in activity 1. So we are actually working on collecting the rumen samples, globally, nationally from Canada. Thank you for Keith, also contribute to the samples from the University of Manitoba Dairy Farm, and we already actually get DNA extracted. And also we are in the process to generate microbiome data. So this is what we are doing right now. So we are collecting all those samples from, you can see here, across Canada. And we are also potentially trying to generate rumen microbial profiles, volatile fatty acid, genome type about 10,000 cattle. We are doing the microbial profiling using short reads of all the samples, long reads about 2,000, and the metagenome metabolome for about 1,000. And then we're going to use those machine learning and to determine the causal effect and also potentially link those causal effects to humans' cultural omics. So this activity also partially supported by BCRC that actually before we get those NSERC program funded, I really appreciate BCRC supported the cattle, beef cattle's initiative looking at the whole microbiome. So where we are right now. So yesterday I talked about in the class, and machine learning to me is cloud and the local. So I really don't know much about it. So I actually took a workshop to understand what is machine learning. So we found machine learning can be a very powerful tool. However, machine learning can be largely biased because most of the algorithm, many cases still based on association, also still based on the data that may not necessarily with high quality and also understanding of the biology. So my postdoc actually used, or in collaboration with a machine learning expert at UBC, we actually adopt a 9 machine learning algorithm and to test which one can potentially use for microbiome research. I'll just give you some brief results and what we found. We found out that human microbiome, we might not need that complex model if we only look at microbiome and the VFA. But if we want to incorporate more phenotypes, that we need to be carefully select which phenotype it's likely to be highly predictable or used for machine learning analysis. So this is what we found, that FCR phenotype is more it's better that to be used for the prediction compared to R5. So we are still trying to solve certain issues about the models overfitting and also the underfitting as well. So hopefully by next year, with more data has been generated, we should be able to develop a better mechanism. Second activity, which is involved in the activity 2 about the human microbiome manipulation led by human, we're actually working on RNAi technology. So what is the idea? Basically, microbes also have the non-coding RNA, and those non-coding RNA can regulate their growth and gene expression. So the idea is more like a human gene therapy. If we can define or design an interference RNA, and we can feed to the cattle, and potentially we can inhibit targeted microbial kind of activity. And this project also is supported by actually RADA, Alberta Beef Producer, and the BCRC. So I want to show some very brief results, and it takes almost 5 years to get here because we are lack of facility to grow methanogen. So I really appreciate now I saw humans culture chamber, and I'm very excited potentially we can also continue to grow our methanogens at U of M. So here is we actually found a non-coding RNAs that in the methanogen genome, and we actually selected non-coding RNAs that specific target MCRA, which is the methyl reductase, that is the key enzymes involved in methanogenesis. And we actually designed the antisense RNA. So this is a very recent result. We can see the growth has been changed when we use different dosage of the antisense RNA. And you can see here the methane significantly reduced. So this is the 0 with the dosage change. So this is the first evidence. I only just get this before I came here. So I'm very excited. There is the first evidence our idea actually worked. So hopefully we all have more results soon that we can actually select the most effective antisense RNA to reduce methane activity. The last one, there are two more activities I want to talk is about the grazing. So we actually now collecting large data set from a grazing cattle that from University of Alberta that we actually looking at how the rumen microbes shift through the grazing and how they can be related to the feed efficiency and methane. So what is interesting? This is how we all microbiome researchers do, forget the chart. So this is very common. We do look at microbial profiles and say, oh, they're different, they shift. So here you can see, seems there is a separation. And then you always look, doesn't matter. So this is important. When you look at all the microbes merged, the metabolites are clearly separated. So this is really raised a very key research question is, when we look at human microbiome based on composition, based on metagenome, it may not lead to the better understanding of the microbial fermentation metabolism activities. So based on those research, we're able to find how potentially microbial metabolites shift during grazing and which metabolites are likely different between the low and high R5 animals and what are those metabolites. So in collaboration with Professor Liang Li at the U of A, we are now developing a rapid metabolomic test. We can potentially identify 900 metabolites with one panel. Then we can potentially use them to screen animals with very specific human microbial metabolites. The last one I want to share the story we have been working on is to really understand 3-NOP. We all know 3-NOP now, that 3-NOP is proved by many governments globally, and 3-NOP are known to reduce methane. So we actually now look at how does 3-NOP work? Does it work for all the cattle sectors? So we actually generate this data almost 8 years ago. Finally, we had the chance to revisit data, understanding what are the key mechanisms of 3-NOP. So I don't have time to go through the details, but the conclusion, you'll be surprised. 3-NOP's mechanism on the rumen microbial impact differs between beef and dairy. So one example, for example, 3-NOP will affect propionate-producing organisms in the beef sector, but is a butyrate-producing organism in the dairy. And also, they may potentially have different metabolism. You can see here the formate in the beef sector, and here we can see there is a butyrate and also hydrogen, directly hydrogen-related metabolism. Second key message, that when you look at the dairy, the 3-NOP effect on the rumen microbiome also differ when the cow is eating different diet. So their efficacy can be potentially mixed or interact with what they eat. So those information potentially give us the idea if we want to give any feed additives, we really need to think about how the rumen microbes may respond to the interaction between feed additives and the dietary type. So you can see here, we can see when it's high grain, the main pathway and the methanogen involved are different from potentially the organisms when they're under the high forage. So those are some of the kind of mechanisms. We know this is a lot of mechanism. You may ask, so what? This is how we can potentially provide fundamental scientific knowledge, say why this cow had 3-NOP, it didn't work because of diet, because of genetics. So you may ask, are we there yet? If you look at path and popularity about research of microbiome, it's always the question, are we there yet? So you can see here we have been generating lots of data, but is it enough? It's not, okay? Because all the rumen microbiome research we do is just a snapshot. Microbiome change all the time, through the day, doing different feeding, and so on. So how we can potentially dissect those dynamic shift, consistent change, and also who are the true colonizer and who are just passenger, but also how potentially microbes interact with diet and nutrition, and also feed additives, that dynamics and the long-term, short-term consequence we don't know. The second one is the most of the research based on metagenome is computation-based bioinformatics. And we are missing real biology. So that's why, again, all the research now at here, University of Manitoba, it's so important. That is our future, because we can always go back to the microbiology. We can validate we found from metagenome bioinformatics. That's the most powerful tool we can use to support our industry. Another one is, as an animal scientist, how we can potentially do in the life cycle, production, and health traits of the rumen microbiome still missing, and how we can potentially identify through the birth and through the production cycle of the dairy cows or the beef cattle, how potentially host together with microbiome regulate its performance. So the take-home message, as a scientist, we still generate more data, but also in the meantime, we need to work with industry directly to address industry's challenge, problems, but also potentially we need all our researchers to work together, collaborate, and also potentially contribute the same goal. So we want to potentially really deliver for this project that we can, again, to design or deliver microbiome intervention solutions that can be effective and to regulate endogenous microbiome and host genetics and potentially reach our 3R goal. So I thank all the funders. I can't just list all of them. They all have supported my research program. Without funding, I cannot be here. Secondly, I thank all the collaborators, not about just many projects, especially this UNSUG team. Everyone is amazing, and I always learn so much from them. And they were also very supportive, and also all the international partners, and also my students from University of Alberta. Now this is my team at UBC. So I think, and thank you all to be here. Thank you.
Thank you, Leluo. As always, great presentation, lots of interesting data. Thank you so much for sharing with us. So I also want to apologize to the online audience. There was a delay for us to be able to connect online. There was a central internet Wi-Fi issue at the university, so I do apologize, but we were able to connect, by a few minute delays here. So I'm going to open the room and also the online forum for the questions. I'm going to start with one. So there is growing interest in genetic selection, breeding for high feed efficiency, for low methane emission. And now there is growing interest to integrate microbiome data, as you mentioned. So, and then you mentioned about the heritability of the microbiome. So I'm always curious to know, what is your opinion about what are the ways that the calves, they inherit the microbiome through the dams, especially when we talk about the dairy environment, because the growing fetus is growing in a sterile environment, and then for the dairy calves especially, we are separating them from the cows right away. So what are those ways that the microbes could be inherited?
Yeah, I think that's a really good question, and there are different theories about it. So one of the theory is the vertical transmission. So basic is from calves, from mom, doing that virginal tract through the birth. Secondly is the potential, we know there's microbes, we call them microbial seeding. So there is a potential microbial signals. They can be even circulated during the fetal stage that potentially our calves are born, or even the infants are born, the host potentially to select because they were reprogrammed. So they can select microbes, potentially have those microbial seeding signals.
Great.
And we don't know, it's just a hypothesis. But most of the critical side, we talk about heritable, again, I was going to write a short communication to correct the term we use, heritable, because it's not the true heritable. It's really host SNP associated. I think it's really how host, through the mechanisms, including defensing, including antimicrobial peptides, including their mucus development, how to select microbes that benefit host the most.
Great, thank you. So any questions from the class? Case?
A very good, interesting talk. one question. What is the rumen microbiome? You showed that the rumen is a very big place. So what is the rumen microbiome? Where should we take the sample from? And how?
I think, Keith, that's a most important question about technical, okay? So actually, we did a lot of efforts to understand how, when to sample rumen, okay? So in the cannulated cows is the best scenario. We actually, in collaboration with Dr. Oba a long time ago, we actually collect rumen samples from 5 locations, cannulated cow, and every 3 hours. And then we look at the microbial composition and diversity, and also where the location different, or is the time where to. So when to collect samples, how to collect samples depends on ideal situation, right? So for beef cattle, we cannot, cannulate all of them. We use stomach tubing. So we also compared cannulated animals with tubing. We didn't publish, but we did all those kind of a technical comparison, how tubing and cannulated cows, their rumen microbiome may different. So we do find what happens is in the rumen, the diversity is not that different among 5 locations because it's the same rumen, they mix, right? But it's the population. So the same bacteria, their abundance may different depends on where location. So if we only interest in diversity, presence, absence, or particular organisms, it is not that critical. But if we want to know activity, that's important. That's why in most of metagenome research, we actually pool 5 locations for metagenome analysis. And the other one is tubing. Tubing and the cannulation depends on how good you're tubing. It also can be up to 90% similarity about diversity or the presence, absence of the textile.
Important location in the rumen? Is it the fiber mat, reticulum? Is it the frontal sac, caudal sac? You could argue that in the fiber mat is maybe most critical.
Yeah.
Because that's where fiber digestion, fermentation takes place. But how do you collect representative rumen fluid? Do you take the digester and you squeeze it? Or,
No, what we do for all the slaughtered beef cows or cattle, not cows, steers, we collect 5 locations, we mix them, and we grind them into the powder under liquid nitrogen, and we take powder for DNA, RNA extraction. So we try to make them representative. To answer your question, which part is more important? I actually don't know. We actually do have samples collected from different locations, but we never did metagenome at the time. I assume because of their diversity, it's similar, and it's only the abundance population that must have a different contribution to VFA. But then if you think about the rumen movement, it depends on when, right? Because we know that fiber matter sometimes, through the day, after feeding, it can be also very different, right? Because we even know protozoa, they're swimming into the bottom, depends on, after how many hours feeding. So that all potentially lead to a critical impact on rumen microbiome research. However, if we standardize sampling protocol at the same time for the same herd, we can potentially minimize those variables. We can only try as much as we can.
Great, thank you. Larissa.
I have kind of 2 questions in one for you, because you had mentioned that 3-NOP affects both beef and dairy differently. So I'm just wondering if we can assume that then any other additive or probiotic would affect beef and dairy differently, and then also if you think there would be any difference between breeds within either dairy or beef. So like if Holstein would have a different reaction comparative to Jersey, for example.
So to answer your second question, there are actually quite some research already done to looking at the breed effect. So there's actually microbiome, not say compared to rumen microbiome between Holstein and the Jersey, I think led by a US group a couple of years ago. So there's evidence different breed will have different rumen microbiome. Second, beef and dairy, we know beef has much more diverse genetics, especially hybrid, versus dairy. When we look at Holstein, you know, when you look at the microbiome within the dairy herd, they have very high similarity. While beef is actually very low similarity. Depends on their, you know, whether they were purebred or genetics. But we do have evidence that the Angus, Charolais, and the hybrid actually have very different microbiota. So because they have very different microbiota, they will respond to the feed additives differently.
Okay, great. So we have time for one more question. There's an online question, sorry.
This question comes from Pasayo. What is good bacteria? Because same bacteria have positive and negative functions.
That's a really good question. And so the good bacteria is usually, I think, we try to determine their roles. And we all know E. coli can be good in our gut if they are part of a commensa, but they can become pathogenic if the gut environment change. So we can't really define the bacteria good or bad. I think really we need to know their function on whether they contribute to the host positively or detrimentally. Even the same probiotics, they can be potentially benefit me, or maybe benefit human, but not benefit me. Because we think they were good, but they're only good for human, but not good for me. So this is what we call how individualize the genetics, individualize the microbiome to potentially welcome or kill the even probiotics we think they were good. So there's no absolute good bacteria.
Great,
to my perspective.
So there are still more questions, but unfortunately, in the interest of time, I need to call the session. There is another class. But thank you, everyone, for joining. Thank you, Dr. Guan, for accepting our invitation and giving such an excellent talk. Thank you very much.
Thank you, everyone.